Imagine a European robin. It weighs less than a smartphone charger, its brain is the size of a pea, and every autumn, it embarks on an impossible journey. Under the cover of darkness, it flies from the forests of Scandinavia, across open seas and vast landscapes, to the warmth of the Mediterranean, thousands of kilometers away. It doesn't have a map. It doesn't have a GPS. Yet, it navigates with pinpoint accuracy. How? Now, think smaller. Think inside your own body. Right now, as you watch this, trillions of chemical reactions are firing in every one of your cells. They are building, repairing, and powering you with a speed and precision that makes our most advanced factories look primitive. How does this microscopic dance happen fast enough to keep you alive? These feel like miracles of biology, the elegant solutions of evolution. But tonight, we are asking a much bolder, much stranger question. Are these miracles powered by the weirdest, most counterintuitive rules in the known universe? The wager on the table is this: is life hacking quantum mechanics? Is it using spooky effects like entanglement, tunneling, and superposition, not in a sterile laboratory at absolute zero, but inside the warm, wet, chaotic environment of a living cell? Tonight, we are not here to amplify the hype. We are here to conduct a rigorous audit of the evidence. Welcome to the Science Audit. Before we can investigate, we need a shared language. The quantum world is notoriously bizarre, but the concepts life might be using are surprisingly tangible when you use the right analogies. So let's build our toolkit. First, let's talk about spin. Forget what you know about spinning tops. A quantum spin is an intrinsic property of a particle, like its charge or mass. It's best to think of it as a tiny internal compass needle. It can point up, or it can point down. North or south. That's it. But here's the first quantum twist. In our world, a compass points one way. In the quantum world, until you measure it, that electron's spin can be in a state of superposition, pointing both up and down at the same time. It's like a coin, spinning in the air. It's either heads nor tails. It's a blur of both possibilities. Only when it lands, when we measure it, does it choose a state. Superposition is the reality of maybe, the existence of multiple possibilities at once. This leads directly to our second concept, coherence. If superposition is one particle being in two states at once, coherence is when multiple particles link their superpositions together. They synchronize their possibilities. Think of it like a choir. Superposition is one singer holding a note that is somehow both a C and a G. Coherence is the entire choir doing it, their strange, multi-tone notes, rising and falling in perfect, harmonious time. This quantum hum is incredibly delicate. A single stray noise can make the choir fall out of sync. That's a process we'll talk about later called decoherence. Third, we have tunneling. This is the ultimate quantum shortcut. Imagine you're a tiny particle, and you're facing an energy barrier, a steep hill you don't have enough energy to climb. In our classical world, that's the end of the story. You're stuck. But a quantum particle also behaves like a wave. Its position isn't a single point, but a cloud of probability. And sometimes, the edge of that probability cloud can leak through the barrier. When that happens, there's a non-zero chance the particle will simply vanish from one side and reappear on the other. It didn't climb the hill, it tunneled through the fabric of reality itself. It's the ghost move. And finally, the most famous and perhaps most profound concept, entanglement. Einstein called it spooky action at a distance. Imagine you create two particles together in a special way, let's say two electrons. Their spins are linked. One must be spin up, the other must be spin down. That's the rule of their creation. Now you separate them. You can send one to the other side of the galaxy. The moment you measure the spin of your local electron and find that it's up, you know, instantly, with 100% certainty, that the other electron, light years away, has just become down. Information seems to pass between them faster than the speed of light. They are no longer two separate things, they are two parts of a single quantum system, a unified whole, connected by an invisible thread that defies space and time. Superposition, coherence, tunneling, and entanglement. These are the tools of the quantum world. Now let's audit the four most compelling cases where biology might be putting them to work. Our first and strongest case begins with that migrating robin in the dead of night. For a long time, the leading hypothesis for its navigation was simple and intuitive: maybe it has a literal compass. Scientists proposed that birds might have tiny magnetic particles, like iron filings, in their beaks or inner ears, physically aligning with the Earth's magnetic field like a compass needle. The problem is, after decades of searching, the evidence for a primary magnetic sense based on iron particles is inconclusive and hotly debated. It doesn't seem to be the whole story. This led to a radical new proposal, born from chemistry in the 1970s and championed by scientists like Klaus Schulten. The idea? The entire compass isn't a physical needle, but a chemical reaction. A reaction whose outcome is determined by quantum mechanics. The entire mechanism, they proposed, lives inside a single molecule in the bird's eye, a protein called cryptochrome. Cryptochromes are ancient proteins found throughout the animal and plant kingdoms. They are involved in regulating circadian rhythms, the internal body clock. But in birds, they seem to have been co-opted for something extraordinary. Here is the proposed mechanism, the radical pair mechanism, step by step. Step 1. A photon of blue light from the evening sky enters the robin's eye and strikes a specific cryptochrome molecule located in its retina. This photon isn't just for vision, it's a starter's pistol. It provides a jolt of energy. Step 2. This energy boost kicks an electron from one part of the molecule to a nearby partner, a molecule called FAD. This act of separation creates what's known as a radical pair. A radical is just a molecule with an unpaired electron, making it highly reactive. We now have two molecules, each with a lonely electron. Step 3. The quantum core. This is where it gets weird. These two electrons, born from the same photon-driven event, are now quantum entangled. Their spins are perfectly correlated. For a brief moment, they form a single, unified quantum system. They are our two coins, linked by that invisible thread. Their combined spin state can be one of two types: a singlet state, where the spins are antiparallel, one up, one down, or a triplet state, where they are parallel, both up or both down. They start in the singlet state. Step 4. The Earth's whisper. Now, the Earth's magnetic field enters the scene. Remember how weak it is? About 50 microtesla. It's far too weak to shove a molecule around. But it's just strong enough to whisper to the electron spins. This magnetic whisper causes the spins to wobble, or precess, at a specific frequency. Crucially, the angle of the Earth's magnetic field relative to the bird's head changes the nature of this wobble. Step 5. The dance of probabilities. This magnetic wobble influences the dance between the singlet and triplet states. The two entangled electrons oscillate back and forth between these two configurations. The Earth's magnetic field acts like a cosmic dial, tuning the speed of this oscillation. If the bird is facing north, the field lines are at one angle, and the electrons spend more time in the singlet state. If the bird turns to face east, the angle changes, and they might spend more time in the triplet state. Step 6. From quantum to neuron. This is the final, genius step. How does the bird's brain read this quantum dance? The answer is chemistry. The chemical fate of the radical pair depends on its spin state. The singlet state and the triplet state lead to different chemical end products. One state might lead to the cryptochrome molecule returning to its ground state, ready for the next photon. The other state might produce a different signaling molecule entirely. The result is breathtaking. The angle of the Earth's magnetic field directly controls the amount of a specific chemical produced in the bird's retina. The bird could literally see the magnetic field of the Earth, perhaps as a pattern of light and shadow, a heads-up display overlaid on its normal vision, telling it which way is north. A quantum compass, built from a single molecule. Okay, that is one of the most beautiful and complex theories in all of biology. But an audit requires cold, hard evidence. What have we got? The evidence is remarkably strong and comes from multiple angles. First, there's the light dependency. Bird navigation is known to depend on light. In total darkness, they resort to other cues. More specifically, it works under blue or green light, but gets disrupted by red light, which is exactly what you'd expect if the starting pistol is a blue photon hitting cryptochrome. Second, and this is the smoking gun, is the oscillating field experiment. Scientists placed robins in a controlled environment, and found they could orient themselves perfectly with the Earth's magnetic field. Then, they bathed the birds in an additional, extremely weak, oscillating magnetic field, a radio wave. When the frequency of this radio wave was tuned to the exact resonant frequency of the electron spins, the so-called Larmor frequency, the bird's sense of direction vanished. They were completely disoriented. It's like trying to listen to a faint whisper while someone is screaming a single, loud note in your ear. The radio field was jamming the quantum signal. Turn off the field, or change its frequency, and their navigational ability snapped right back. This is incredibly specific and hard to explain with any classical model. Third, the hardware has been identified. Molecular biologists have located a specific version of cryptochrome, called Cry4A, in the retinas of migratory birds, in the exact cells, the double-cone photoreceptors, that would be best placed for this job. The levels of this protein even increase during migratory season. The verdict? The case for the avian quantum compass is very strong. It is the leading theory by a wide margin. While we haven't yet managed to film the radical pair flipping between singlet and triplet states inside a living, flying bird, the circumstantial evidence is overwhelming. The mechanism makes specific, testable predictions, and so far, it has passed those tests with flying colors. The ledger is heavily weighted toward yes. Our next case moves from the vastness of the sky to the microscopic universe within you. Your body is running on chemical reactions. To be precise, something like 37,000 billion chemical reactions are happening in your body every single second. And many of them face a fundamental problem: speed. At the balmy 37 degrees Celsius of your body, most of these reactions should be incredibly slow. The molecules involved need a significant jolt of energy to get over an activation hill before they can react. The heroes that solve this problem are enzymes. Enzymes are giant protein molecules, folded into intricate 3D shapes. They act as molecular matchmakers. They grab the reacting molecules, the substrates, and hold them in a perfect orientation within a special pocket called the active site. This precise positioning dramatically lowers the energy hill the reaction needs to climb. The classic analogy is a lock and a key. But sometimes, even this isn't enough. For some of the most fundamental reactions in life, there's a quantum plot twist. The enzyme doesn't just lower the hill, it helps a key player, often a hydrogen atom, to not bother climbing it at all. It encourages it to tunnel straight through. Why hydrogen? Because it is the lightest element in the universe, consisting of just a single proton. Its lightness means its quantum nature, its wave-particle duality, is much more pronounced than for any other atom. Its position is fuzzier. This fuzziness is what allows it to tunnel. This is a fascinating idea. Life using the ultimate ghost move to speed things up. But again, how could you possibly prove that a single atom is quantum tunneling inside the chaotic, jiggling environment of an enzyme? You perform one of the most elegant experiments in biophysics. You use a heavy imposter. You take the normal hydrogen atom in your substrate, and you replace it with its stable isotope, deuterium. Deuterium is chemically identical to hydrogen. It has the same charge, fits in the same active site, and forms the same bonds. But it has a neutron in its nucleus along with the proton, making it twice as heavy. To an enzyme's active site, it looks and feels the same. But to the laws of quantum mechanics, its heaviness makes it vastly worse at tunneling. It's like asking a ghost to wear a lead suit. Its probability wave is much more localized. So you set up the experiment. You measure the rate of the reaction with the normal, hydrogen-containing molecule. Then you measure it with the deuterium-swapped molecule. If the reaction were purely classical, if the proton had to physically climb the energy hill, making it twice as heavy should slow the reaction down by a predictable, modest amount. But if tunneling is the main shortcut, the results should be dramatic. By blocking the tunneling pathway with the heavy deuterium, the reaction should slow down enormously. This is exactly what scientists find. In many enzymes, they observe what is called a large kinetic isotope effect, or KIE. The deuterium-powered reaction can be hundreds, or even thousands, of times slower than the hydrogen-powered one. These numbers are impossible to explain with classical chemistry alone. It is the smoking gun for quantum tunneling. But it gets even better. Further studies on the temperature dependence of these reactions revealed something incredible. The enzyme isn't just a passive stage for this quantum event. The giant protein molecule is constantly in motion, breathing and flexing. It turns out that these motions are perfectly choreographed. The protein flexes in a way that, for a fleeting moment, squeezes the energy barrier, making it momentarily thinner and lower. It's in that precise instant that the hydrogen atom makes its quantum leap. It's a breathtakingly beautiful dance between the large-scale, classical motion of the protein and the purely quantum jump of the proton it holds. Life isn't just allowing tunneling to happen, it has evolved to actively promote and time it. The verdict? The evidence for quantum tunneling in many enzymes is persuasive and widely accepted. It's not a speculative idea, it's a core part of how we understand many biochemical reactions, particularly those involving the transfer of hydrogen. It is a clear case where evolution has stumbled upon a quantum phenomenon and exploited it for a critical biological advantage: speed. Our third case takes us to the silent, green engine of our planet, photosynthesis. It is the single most important biological process on Earth. It is the foundation of our food webs and the source of the oxygen we breathe. And its success hinges on a problem of efficiency. When a photon from the sun strikes a leaf, its energy is captured by an antenna molecule, usually chlorophyll. But this is just the first step. That captured energy, now in the form of a quasi-particle called an exciton, must be transported to a very specific destination called a reaction center. It's only in the reaction center that the sun's energy can be safely converted into chemical energy. The antenna system is a vast, dense forest of pigment molecules. The reaction center is a single, small clearing in that forest. If the exciton were to just wander around randomly, bouncing from molecule to molecule like a drunk stumbling through the woods, it would likely lose its energy as heat long before it found the clearing. Yet, in many photosynthetic systems, this energy transfer process is over 95% efficient. How does the energy find the fastest path, almost every single time? The quantum hypothesis is as elegant as it is mind-bending. It suggests that the exciton doesn't have to choose a single path. Thanks to superposition and coherence, the energy can, for a few brief moments, behave like a wave and explore all possible paths simultaneously. Imagine dropping a pebble into a pond. The ripple expands in all directions at once. The quantum theory of photosynthesis suggests the exciton does something similar. It samples the entire network of chlorophyll molecules at once, instantly finding the most direct route to the reaction center. It's not a search, it's a wave-like propagation that collapses onto the most efficient path. In the early 2000s, this idea went from theory to tantalizing evidence. Using incredibly fast laser pulses, flashes of light lasting just femtoseconds, research groups, like that of Graham Fleming at UC Berkeley, zapped these photosynthetic complexes and listened for the echo. What they found stunned the scientific world. They detected long-lasting, oscillating signals, quantum beats. It looked as though the excitons were remaining in a coherent, wave-like state for hundreds of femtoseconds. This was far longer than anyone thought possible in a warm, wet biological system. For a time, it seemed like slam-dunk evidence that plants were using quantum coherence. But science is a self-correcting process. An audit demands we look at the counterarguments. Is this really a quantum search algorithm, or are we misinterpreting the music? This is where the scientific debate is currently raging. Other research groups began to question the initial interpretation. Could the powerful lasers used in the experiments actually be creating the very coherence they were measuring? Or, more subtly, were these beats not purely a sign of electronic coherence, but of something else? The leading alternative idea is that we are seeing vibronic coherence. This is a hybrid state where the quantum electronic state of the exciton couples with the natural, classical vibrations of the molecular structure. Think of it like a perfectly timed push on a swing. The jiggling of the protein scaffold isn't just noise, it's structured vibrations. And these vibrations might be locking arms with the exciton, helping to wash it downhill towards the reaction center. So, is it quantum? Yes, but perhaps in a more subtle, messy, and robust way than the initial, pure coherence model suggested. The debate now is about the precise nature of this quantum effect and how much of a role it really plays. Is it the essential secret to efficiency, or just a small contributing factor? The verdict? The community is currently divided, but the consensus is shifting. The initial idea of a long-lived, purely electronic quantum search is now seen as too simplistic. However, the evidence for some form of transient, vibrationally assisted quantum coherence remains strong. It's likely that evolution has crafted a system that uses a good enough quantum trick, leveraging the surrounding molecular noise instead of fighting it, to gain an edge in efficiency. The case is not closed, but the investigation has opened a fascinating new window into the physics of life. Our final case is by far the most controversial. It concerns one of our most primitive and evocative senses, olfaction, the sense of smell. The textbook explanation for how we smell is a theory called shape theory or odor and receptor theory. It's beautifully simple. Molecules with a certain smell, say, vanilla, have a specific 3D shape. In your nose, you have millions of olfactory receptors. When a vanilla molecule floats by, it fits perfectly into a vanilla-shaped receptor, like a key into a lock. This binding triggers a signal to your brain, and you perceive the smell of vanilla. This theory explains a lot, but it has some nagging problems. For example, there are molecules called stereoisomers that are mirror images of each other. They have the exact same atoms and the same shape, just left-handed and right-handed versions. According to shape theory, they should smell the same. But they often don't. One version of the molecule carvone smells like spearmint. Its mirror image smells like caraway. Why? This led biophysicist Luca Turin to resurrect and champion a radical alternative in the 1990s, the vibration theory of olfaction. His proposal? Our smell receptors don't just detect shape, they detect the vibration of a molecule's chemical bonds. The theory goes like this. When an odor molecule is docked in a receptor, an electron on one side of the receptor wants to get to the other side. But it's blocked by an energy gap. Turin proposed that the electron can tunnel across this gap, but only if the docked odor molecule can absorb the excess energy. It does this by vibrating its bonds at a very specific frequency. It's like a toll booth on a quantum highway. The electron is a car. The receptor is the booth. To get through, the car needs to pay a toll of a very specific amount of energy. The odor molecule is like a passenger that can pay the toll, but only by playing a specific musical note, its vibrational frequency. If the molecule plays the right note, the electron tunnels, a signal is sent, and you perceive a smell. In this model, your nose is not a collection of locks, it's a collection of tiny, nanoscale vibration spectrometers. It's a stunningly creative idea, but an audit lives and dies on reproducible evidence. What does the data say? This is where the story gets messy. Turin and his colleagues published studies that seem to support the theory. They trained fruit flies to distinguish between two molecules that were identical in shape but had different vibrations because normal hydrogen atoms were swapped for heavy deuterium. The flies, they claimed, could tell the difference. Similar results were reported in some human studies. However, the core principle of science is reproducibility. And many other, often larger, labs around the world have tried to replicate these key experiments, and have failed. They found that neither flies nor humans could reliably distinguish between the deuterated and non-deuterated molecules. The scientific community largely remains deeply skeptical, pointing out that the shape theory, while not perfect, can be refined to explain many of the anomalies that the vibration theory claims to solve. The verdict? The vibration theory of smell is a fascinating, compelling, and beautifully unorthodox idea. It makes testable predictions, which makes it good science. However, at present, it remains an outlier. The evidence in its favor is weak and contested, while the counter-evidence is significant. The audit must conclude that, as of today, this case is not proven. The lock and key model, in some form, remains the dominant explanation. So, after our deep audit, where do we stand? A likely quantum compass in birds. A persuasive case for tunneling in our enzymes. A tantalizing and complex quantum story unfolding in photosynthesis. And a beautiful, but unproven, idea about our sense of smell. The pattern is clear. This isn't quantum mechanics everywhere. Life is pragmatic. It's a tinkerer, not a grand theorist. It uses what works. The central objection to all of quantum biology has always been the environment. The warm, wet, chaotic world of a cell should be a quantum killer. Any delicate coherent state should be destroyed by random collisions in femtoseconds. This destruction is called decoherence, the applause that drowns out the quantum whisper. But perhaps we've been thinking about noise all wrong. Maybe noise isn't always the enemy. Recent theories suggest that in some systems, like photosynthesis, the structured vibrations of the surrounding environment, the noise, might actually protect quantum coherence and help guide energy transfer. It's a phenomenon called environment-assisted quantum transport. Life might not be silencing the noise, it might be conducting it. It has learned to time the quantum whisper to land in the brief moment of quiet before the applause returns. If this view is correct, it fundamentally reframes our definition of life. Life isn't just complex chemistry fighting against the forces of physics. It is chemistry that has learned to bend the deepest rules of physics to its will. It operates at the boundary, a masterful conductor of energy and information, using every trick in the book, classical and quantum. This isn't just a fascinating philosophical insight. It has profound practical implications for our future. If we can truly understand the radical pair mechanism, we could engineer novel chemical magnetometers that operate at room temperature, revolutionizing navigation and medical imaging. If we can master the principles of enzyme tunneling, we could design new catalysts that perform clean, efficient chemistry, helping to solve our energy crisis. If we can reverse engineer the secrets of photosynthesis, we could build solar energy collectors with near-perfect efficiency. We are standing at the dawn of a new science. One that blurs the lines between physics, chemistry, and biology. It shows us that in every flying bird, in every blade of grass, and in every beat of our own hearts, there may be a whisper of the quantum world. A hidden layer of reality that life discovered billions of years ago, and that we are only just beginning to understand. And that's the core mission of science, to keep peeling back the layers, following the evidence no matter how strange the destination. If this deep audit sparked your curiosity, you're going to love the video waiting for you on the end screen. Go ahead and choose your next adventure into the frontiers of knowledge. To ensure you don't miss our future investigations, make sure to subscribe and activate notifications. We go deep like this every single week. Finally, I have a question for you. Of these 4 incredible ideas, the bird's quantum compass, the tunneling enzymes in your cells, the coherent garden of photosynthesis, or the vibrational theory of smell, which one blew your mind the most? Drop your answer in the comments below. Let's see which concept wins the popular vote. Thank you for joining the audit.